So, dear Professor Molnár, thank you for joining us today. It's a real honor to have you in our e-lecture series. And thank you for everybody for attending this lecture. So I will just start with as much brief as I can introduction to Dr. Molnár's biography. So, and I hope I'm pronouncing it correctly. It's Molnár.
Absolutely
right?
perfect.
Yeah, thank you. So, Professor Molnár is the professor of developmental neuroscience at the University of Oxford. He earned his MD at the Albert Zent Georgi Medical University at Szeged, Hungary, where he started his residency in neurological surgery until he moved to Oxford in 1989. He earned his doctorate in philosophy at the University Laboratory of Physiology in the laboratory of Professor Sir Colin Blakemore, studying the multiple mechanisms in the establishment of thalamocortical innervation. He continued his work on cerebral cortical development at Oxford as an MRC training fellow and junior research fellow at Merton College. He also investigated thalamocortical development working with Professor Egbert Welker at the Institut de Biologie Cellulaire et Morphologie, Université de Lausanne, Switzerland, and learned optical recording techniques to understand early functional thalamocortical interactions in the laboratory of Professor Toyama at Kyoto Prefectural School of Medicine in Japan. He was appointed to a university lecturer position at the Department of Human Anatomy and Genetics, associated with a tutorship at St. John's College, Oxford from 2000, and received the title Professor of Developmental Neuroscience in 2007. So Dr. Molnár is known for his key contributions to our understanding of how the birth of cortical neurons is regulated, how they migrate, differentiate, generate axons, and how the thalamocortical projections are guided to the cerebral cortex, and how the thalamic afferents interact with the earliest transient cortical circuits. He pioneered the identification of layer and cell type specific markers, including the earlier larger transient subplate neurons. He described stage-specific intra and extracortical integration of the subplate neurons during development and discovered a new type of corticothalamocortical circuit established by layer 6B neurons. His research revealed how thalamic connectivity shapes the developing cortex and how the cortex regulates its own state during sensory perception and sleep. By examining and comparing these mechanisms in various species, including humans, he showed how specific alterations of developmental mechanisms at the pallial and subpallial boundary and in the cortical germinal zones have led to the evolution of the mammalian cerebral cortex. Again, Professor Molnár, it's an honor to have you. And to all our friends watching us now, please note your questions in the Q&A or chat area, and we will address them either during the presentation or at the end of Professor Molnár's presentation. Thank you again, and it's an honor to have you.
Thank you very much, Maria. I have very little to say after this introduction because you actually went through my lecture pretty much. So it's a great honor to be here and talk to you for the Cyprus Biological Society Excellence in Biology. And I have to start with some kind of historic background because this is a very special year this year in Oxford. So it is a special year because it's the 400th anniversary of the birth of the person who gave the term neurology or neurology to the word. And this is Thomas Willis. And Thomas Willis was born in this house in 1621. It's about 30 minutes drive from Oxford in East Bedwin. And he moved to Oxford with the family and eventually started working in this street, which changed very little over the last 400 years. This is Merton Street. And you can see on the street here, Corpus Christi College. And then you see one of the oldest colleges, Merton College Chapel here. Basically his house was opposite to the entrance of Merton College. You can see now the Porter's Lodge So this is still Merton College. And opposite to this, so if you come to Oxford, you have to walk through the street. This is the house where he worked, and he coined the term neurology or neurologia. So we should remember Willis because of this this year. So he had some other contributions, of course, much bigger contributions to this. And of course, every medical student have to remember the name of Willis because of this arteriosus circle at the base of the brain, which is formed from the two internal carotid and the two vertebral arteries. But he was not the first to describe this, but he had the best illustrations because of Christopher Wren's drawings. Now, Willis noticed that when he compared brains of different animals and also with human, that it was really the cerebral cortex, which was much bigger than in a sheep, for instance. So he noticed that the brainstem was very similar. You had the Willis circle in both species, and the brainstem had very similar proportions, but the cerebral cortex was much bigger and much more convoluted in human. And because of this, he argued that the cerebrum is the primary seat of the rational soul in man and the sensitive soul in animals. It is the source of movements and ideas. And he basically got it absolutely right. It is the cerebral cortex, which is the seat of our higher cognitive functions, language, episodic memory, reasoning. And he had another line of arguments to suggest that indeed it is the tissue of the cerebral cortex which is involved in these functions. And that was because of his neuropathological insight. So he followed his patients, and when they died, he dissected some of them. And on the right side, you see a brain from a boy who had signs of intractable epilepsy and also learning difficulties. And Willis noticed that the surface of the brain was very different. This brain had more folding, more sulci, more gyri, but a smaller one than a normal individual. And that's why he linked these conditions to the structure of the cerebral cortex. So he was way ahead of his time. In 1664, he proposed this association between the structure of the cerebral cortex and these diseases. And indeed, probably in 1664, he was looking at the brain of a boy with polymicrogyria syndrome. And this syndrome is due to abnormalities in neurogenesis and migration, and this is changing the shape of the brain. You will have smaller, but more frequent sulci and gyri on the surface of the brain. You can have other conditions like lissencephaly when you have a smooth brain, also associated to abnormal brain development, abnormal cortical neuronal migration. So that's why Willis is my hero, because he made this association almost 400 years ago, and he was absolutely right. So now we know that there are several brain developmental abnormalities, and most of these can occur during in utero life, which is the most important period in the brain development. As you all know, our brain is beginning to form from a neural plate, and this plate is going to close into a neural tube, and the rostral end of this tube will form the telencephalic and diencephalic vesicles, and then the brain, the cerebral cortex, will develop from this telencephalic vesicle. And you can see that there is a disproportionately large growth of this part of our brain. And most of our neurons are born with us. My neurons were born in Hungary, and I still have those sets of neurons in my brain, whereas my microglia, astrocytes, endothelial cells, they were probably replaced by now. And also my liver, my gut lining, my skin, they have a replacement of the cells very frequently. But our neurons in the central nervous system, they don't have this capacity, with the exception of very small regions where you still have neurogenesis. So that's why it's so important to study these early steps of development, because then we will have these neurons for the rest of our lives, and it can have an impact on our life and also the life of our family. And some of these cognitive conditions I listed here, they are all developmental abnormalities of or conditions of our brain. So schizophrenia, autism, learning difficulty, or dyslexia is basically a brain developmental condition. So one in 10 of us have dyslexia in the population. Now, all these developmental steps occur mostly in utero. So the neurogenesis, gliogenesis, migration, axonal growth, synaptogenesis, they all start in utero, and even myelination. And some of the cells will actually die off very early on. They are no longer needed later on in the adult brain. And you see that we peaked around the age of 12 when our cerebral cortex was the thickest. After that, there is a decline in the thickness of the cortex, and also decline in cell numbers. So that's why development is so important. In all mammals, we have roughly the same development of our brain. And usually the convention is that we put the ventricular surface down here. So this is close to the cerebrospinal fluid field ventricle. So if we cut the brain in a coronal fashion, then you can see that this is the inside the ventricle. So that's a ventricular surface. And then you have the outside, which is next to the skull, meninges and the skull. So most of the neurons are born here in the ventricular surface. And if I play this time lapse, you can see that most of the divisions occur in the ventricular and subventricular zone. And then these cells will migrate out to the surface, forming the preplate or primordial plexiform zone. And then this first generated cell layer in preplate will split into two parts, to marginal zone and subplate, by the later arriving cortical plate cells. So across almost the entire neural tube, you have the same kind of principles for the neurogenesis, but from now on, I shall be mostly talking about the cerebral cortex. You see that the first generated cells are in the subplate and also in the marginal zone. And my talk will be mostly of this cell layer, this largely transient, early generated cell layer, the subplate cells. And in the first part of my talk, I shall be discussing some of the developmental roles these cells have during embryonic and early postnatal time. And then later on, I shall be asking the question, what do the remnants of these cells do in the adult brain? Because some of these cells will survive into adulthood. The majority of them will die. The estimate is around 80% in ferret and human, but then you will have some leftover cells. And I want to see what is the function of these repurposed cells in the adult brain. So lots of groups had a great contribution to the understanding of the cell layer. The name subplate is coming from Ivica Kostovic and Paško Rakic. Carla Schatz had a huge contribution to the understanding, together with Donna Ferrero, Heiko Luhmann, and Patrick Kennold. So the subplate is huge in the developing brain. And you can see here in a macaque brain that the subplate is actually bigger than the cortical plate. It's full of cells. Sometimes you even have compartmentalization. And then they form a platform on which the cerebral cortex will develop. So you have a dynamic scaffold system, which is helping the construction of the brain, which will develop between the marginal zone and the cortical plate. And eventually these are the cells which will remain. So my talk will be in three parts. First, I just discuss the origin of these early connections in the subplate neurons. Then I look at the transient subplate circuit formation. And then finally, I shall look at the adult remnants of the subplate, the shadows of the subplate, and what do these cells do in the adult brain. So where are subplate cells coming from? Now, there are multiple sources for subplate cells. Most of the subplate cells actually come from the ventricular, subventricular zone, as I showed you on the time-lapse movie, and then they migrate out to the cortex. And some subplate cells, especially the GABAergic subplate cells, will come from the ganglionic eminences, and they migrate tangentially to the subplate. And you also have some populations, mysterious subplate cell populations, which are glutamatergic, and they migrate tangentially to the cerebral cortex from the rostral medial telencephalic wall. So you have multiple origins, just like for the Caja-Rechus cells, which are in the layer 1, they have multiple sources. The subplate cells also have multiple sources, and that makes them so interesting. Now, once they are in position, and again, we are looking at the coronal section of the embryonic brain. You see the cerebral cortex here, then you see the thalamus, and also the ganglionic eminences, the lateral medial ganglionic eminences. Just when these subplate cells arrive to the cortical plate, then they immediately start to grow projections towards the thalamus, and also the thalamus will send projections towards the cortex. And these projections mingle with one another in the internal capsule. And in fact, the thalamocortical projections will get associated to the early corticofugal projections from subplate, and this is how they pass through the paleo-subpaleo boundary to reach the cortex. And in fact, without this interaction, they cannot make it to the cortex. There are lots of developmental abnormalities at that particular point. And after that, the corticothanamic projections will invade the thalamus, and the cortico-subcerebral projections will leave through the cerebral peduncle, and they will grow into the spinal cord, colliculi, and the basal pons. So this is the same algorithm in all mammals, and the role of the subplate was demonstrated when we looked at the Reeler mouse, where the subplate cells are displaced in relation to the cortical plate. So they are no longer below the cortical plate. In the Reeler mutation, the subplate cells are at the surface, and sure enough, the thalamocortical projections were also growing up to the surface from where they were then growing down to the cortical plate. So the position of the subplate is orchestrating the ingrowth of the thalamocortical projections, even in the cortical plate. And this association is very interesting, and it is established in the internal capsule, and then it's going through the cortical plate all the way to the surface. Now, if you have displaced scaffold from the corticofugal projections, so here I label these up with a reporter line in the Gali-Tau EGFP mouse. Basically, these early corticofugal projections cross the paleo-subpaleo boundary, and then the thalamocortical projections co-fasciculate with them and reach the cortex. Now, if there is an alteration in the early corticothalamic projections, then there will be an alteration also in the thalamocortical pathway. So here in the EMX2 knockout, normally the two pathways run in the internal capsule, but if you have a derailed corticothalamic projections which touch down to the bottom of the diencephalic telencephalic boundary, then also the thalamocortical projections will touch down here, and you will have this aberrant root. So all these suggested that there is an intense interaction between the thalamocortical and corticothalamic projections. Now, the role of the subplate is not finished after these thalamic projections reach the cortex. This is actually just the beginning. They establish a transient circuit within the cortex. And let me just show you some key findings in this area. So it has been known a long time ago that the Ivica Kostovic and others described it that the first synapses are formed in the subplate. And these synapses, some of them actually thalamocortical synapses. And we demonstrated that these synapses were functional using voltage sensitive dyes. We cut some sections of brains where you have the thalamus, the segment of the primary somatosensory cortex, and the interlinking pathways. And then we stimulated the thalamus, and we looked at the evoked responses with voltage sensitive dyes. And you can see that most of the responses are in the subplate initially. And in fact, you can follow the activation of the different cortical layers by cutting these sections in different stages. So let me just show you a newborn response. So we have a thalamocortical slice here. We put an electrode into the thalamus, and we are going to stimulate this region after staining the sections with voltage-sensitive dyes. And you can see that after the activation, you have the spread of action potentials to the cortex, and you have a long-lasting sustained depolarization in the lower layers of the cortex. So this is a newborn animal. And if you cut these similar sections earlier, then it's restricted to subplate. And you also see some activation in the striatum. So the response is quite long-lasting and sustained, but it is changing after birth. And let me just show you two examples, one a 2 days old and a 4 days old slice, where we lined up the videos at the same time. So we are going to stimulate the thalamus at the same time. And you can see that the 4 days old response is much faster, and it's columnar in the segment of the primary somatosensory cortex. And when it's already finished in the 4 days old animal, this is when you still have a very intense response in a 2 days old. So just 2 days maturation can make a huge difference in the dynamics of the response, which you can elicit in these cortices by stimulating the thalamus. So there are lots of changes in the cortical circuitry, and several groups contributed to the understanding of these transient circuits, especially Carla Schatz's group, Patrick Kennold, Heiko Luhmann, and also my group together with Simon Butt. And what we could see is that the thalamocortical projections already stimulated subplate cells, which fed this activity to layer 4 from early stages. So the thalamic projections still did not reach into their ultimate target cells in layer 4, but they already stimulated subplate cells. Then they developed their ultimate synapses in layer 4, but there was a transient circuit coexisting with subplate and some somatostatin positive neurons. It was Andre Marcus Smith who described this in Simon Butt and my laboratory. And after these transient circuits, now you have a more permanent circuit, which is now stabilized in the adult, but without subplate, you could not develop these connections. So you need the subplate even after the thalamocortical projections arrive to the cortex. You have a transient circuit which is feeding into and helping the maturation of the ultimate synapses. And then after all this, the majority of the subplate cells will disappear and you have the adult circuit. So they are a transient dynamic scaffold. And if you change the flow of sensory input, you can actually change the assembly and the disassembly of this pathway. Let me just show you an example from my own group. So we used again this Gali-Tau EGFP mouse, where it's a reporter line where the subplate cell bodies and also their neurites, so axons and dendrites, come blue. And you can see that initially the subplate cells, they project into the cortical plate. And then you have this clustering of the subplate input according to the sensory periphery. In the primary somatosensory cortex, you have these barrel fields. And then after the first postnatal week, they get retracted and they only stay in the septa. You can see this much better on this tangential section. So initially these neurites, they target the middle of the cortical plate, and then later on, they move out to the septa. So we asked the question, what's happening if we remove a row of whiskers and we change the flow of the sensory input? And what was very interesting, that actually this delayed the retraction of the neurites from that corresponding row, suggesting that the disassembly of this transient circuitry is actually dependent on the sensory input. So these transient subplate circuits are key for the establishment of the adult circuits. And if there is a delay in establishing this circuit, then more subplate cells survive. So they orchestrate the ingrowth into the cortex. And perhaps there are many other transient cells, perhaps some closer to the thalamus in the thalamic reticular nucleus, which this is the location where the corticofugal projections wait before they grow in to the thalamus. And they might have a similar role as subplate, where the thalamic projections accumulate, and then they form a transient circuit to reach the cortex. But we have much less information of this corticothalamic side of things, but it's an interesting possibility that the thalamic reticular nucleus is the subplate of the thalamus for the corticothalamic fibers. Now, what's happening to these transient circuits after the maturation of these pathways? And I just show this Lauer diagram from Lana Vassung's publications. Lana is making some incredible contribution to the human imaging field, relating it to human histopathology. And so she color-coded the subplate. And you can see that initially there is and red is increase and blue is decrease in numbers of the and thickness of the subplate. And you can see that there is initially an increase in subplate at the opercular cortices, but then there is a drastic decline of the subplate compartments. So subplate cells will disappear from the brain and the cortical plate will grow. Now, this has been known for some time from the work of Marla Luskin and Carla Schatz. So they used birth dating. So they injected thymidine analogs at the time of the birth of these cells in ferret and cat cortices. And they described that early injections resulted in the labeling of the subplate cells, slightly later, layer 6. And if you followed these labeled cells later on, so not at birth, but maybe 3 months of age, then you can see that there was a drastic decline in the number of labeled cells in subplate, where there was not so much decline in layer 6. So, so Marla and Carla suggested that there is a preferential cell death in subplate cells, so they disappear from the postnatal development. And this is very much the same in monkey. So Paško Rakic and Divica Kostovic describe this. And some of these cells which remain from subplate, they are the interstitial white matter cells in a primate. So you have quite a few cells. So in a macaque, you can see that you have a lot of subplate cells initially, and they are even compartmentalized. And then you have only these interstitial white matter cells. And in the adult, you have even less. Now, these are the remnants of the subplate cells. And in a rodent model, you also have some remnants of the subplate cells, but they will form the layer 6B in the mouse. So it's about an 80 micron thick, very thin cell layer. And these contain the remnants of the cells, subplate cells, which you can establish with birth dating. Now, why should we care about these remnants of the subplate cells? Why it is interesting? And I want to convince you that it is a very interesting cell population. First of all, it has great clinical relevance. If you look at schizophrenic brains from patients from schizophrenia, and you look and you plot and you count the number of interstitial white matter cells, then you have a great increase in the frontal cortices. And Akbarian published a lot on this. And these, why is it that we have more subplate cells surviving in these conditions? And they are the shadows of our subplate. So why is it that they survived? And we are very interested in the possibility that if you have desynchronized burst activity or asynchronized activity or reduced activity due to in utero infection or inflammation or drug exposure or hypoxic ischemia, then perhaps the normal cortical development will be altered. And then you will have these persistent interstitial white matter cells, the remnants of the subplate. And then they can cause neurological or psychiatric conditions. And in schizophrenia, you have more of these cells. So we are very interested in the possibility that these cells cause these symptoms. And that's why my group is very interested in understanding the function of these remnants of subplate, these repurposed cells in the adult brain. Some of the characteristics of these cells is very unique. And I now, in the rest of the remaining time of my talk, I will just show you what do we know about the output from these cells, what is the input to these cells, and then we are going to silence these cells, and I look at how this will affect the behavior of the adult animals. So the link between these adult subplate cells and cognitive conditions like schizophrenia or autism was established a long time ago. And we also noticed that if you look at the gene expression patterns in these cells, in the adult mouse brain, then you pull out gene networks which contain a lot of genes which are involved in susceptibility of these conditions, more than any other layer in the cerebral cortex. So that's why we became interested in studying the cell populations which survive in the adult brain. So we combined birth dating with marker expression, and we established when these cells disappear and what are the markers that will reveal the population which will stay in the adult brain. And after this, now we have access through these expressed markers to monitor and modulate these cell populations selectively. So with the knowledge of these genes, now we can express Cre recombinase in some of these cells, and then through this we can actually trace their connections, output, input, and also we can use genetic manipulations to silence or activate them or to actually prevent their cell death. So what is the output, what is the input, and what are the consequences of silencing of these layer 6B populations? For this, I have to get into a little bit in more detail of the thalamocortical anatomy. So as you know, most sensory information is reaching the cortex through the thalamus. And you have two types of thalamic nuclei. Only the first order thalamic nuclei, like laterogeniculate nucleus, ventrobasal complex, or mediogeniculate nucleus, these are the only ones which receive direct sensory input, and then they relay this to the cerebral cortex. The higher order thalamic nuclei, they receive their input from cortical areas, mostly from layer 5 and 6. And then they relay this input back to other cortical areas. So you have a transthalamic cortico-cortical communication through these higher order thalamic nuclei. And layer 5 has a selective input to these higher order thalamic nuclei with very powerful large boutons. And that's why Mary Sherman and Ray Guillory call these the drivers. Now, we were very interested to see what is the position of layer 6B. Is it similar to a layer 5 or 6A? I just make a long story short. Layer 6B is very different from both from 5 and 6A. It's a special pathway. Some of these neurons, they have very selective output to the higher order thalamic nuclei, and they have small boutons, smaller than the driver projections from layer 5. So we used Cre expressing mouse lines, which we crossed with reporter lines. And you can see that most of the cells are here in the lower part of the cortex in layer 6B. And you don't have cells in the thalamus. These are all projections. And you can see that the primary nuclei, like VB, LGN, MGN, they were empty. So these particular cell populations, they selectively innervate the higher order thalamic nuclei. Now, we looked at this with viral tracing, Cre-dependent adeno-associated viral tracing in these Cre lines. And we noticed that there is an area specificity, there is very extensive intracortical connectivity, and there is also IPSI and contralateral projections to the thalamus. And some of these projections even reach the superior colliculus and other midbrain regions. So very extensive connections. But what was very, very interesting is that the pathways did not form projections to the thalamic reticular nucleus. This is similar to layer 5 and in contrast to layer 6, but they did form synapses in the higher order thalamic nuclei like the PO. So we also looked at the size of these boutons, and they are much smaller than the layer 5 driver projections. So we have a third type of pathway, corticothanamic pathway, it seems, and layer 6b is different from layer 5 because they have small boutons, although they also target higher order thalamic nuclei, and they have this selectivity, and it's still, we don't know functionally whether they are drivers or modulators, but some experiments are in progress to determine that. So we discovered a third type of pathway from layer 6b to the thalamus, and this pathway could be very interesting because of several functional aspects. I explain why this might be interesting. For that, I have to show you some data. What is the input to layer 6b? So how can we label the cells which provide input to these cells? And it was Ed Callaway at Salk Institute who developed these methods of monosynaptic rabies tracing. So what they discovered is that you can express rabies receptor in maybe Cree-dependent manner in a selected cell population. And that's what we did with layer 6b. And then you can infect these cells with modified rabies virus, which will jump one synapse. And after this, you have a very good idea where are your starter cells and where are your cells which project to these cells. You can do it in a cell population or an individual cell group or an individual cell. Actually, we use the group at the moment for this. So who is talking to our layer 6b cells? So these are our starter cells in layer 6b, and you can see lots of layer 5 cells communicate with layer 6b cells. And also you have some extensive input from the thalamus, and also what we were really surprised to see, that you also had some lateral hypothalamic input to layer 6b cells. So why am I so excited about the lateral hypothalamic input? I'm excited because some of these lateral hypothalamic projections are actually orexin positive. Orexin is a peptide which is produced just in the lateral hypothalamus, and this peptide has a major importance in the maintaining arousal. And if you don't have orexin, you can have cataplexia. You can suddenly fall asleep. Perhaps when you get very excited, you hear a joke, then if you don't have orexin, you can fall asleep. Or you have these videos on YouTube on dogs with mutations of orexin or the receptor. So that's why we are so interested in this direct lateral hypothalamic input, and which is targeting, for instance, here it's a prefrontal injection, and from here you have extensive cortical and also input to thalamic input to these cells, and also some from the lateral hypothalamus. So and some of these labeled cells in the lateral hypothalamus were actually hypocretin or orexin immunoreactive. So this is a very exciting possibility that orexin is acting on directly on the cortex and also directly on the higher order thalamic nuclei. So because of this, they are in a strategic position to regulate these particular pathways, which are so important for some of the higher cognitive functions. So it has been known for a while from the work of Bayer and colleagues that it's only layer 6B which have the sensitivity to orexin in the cerebral cortex. The other cells don't have orexin sensitivity. And also higher order thalamic nuclei, they show more sensitivity to orexin than the first order thalamic nuclei. So we wanted to test the idea that if we change the activity of layer 6B cells, then we can actually change the state of arousal or sleep of the animal. So for that, we decided to silence layer 6B neurons directly, but don't touch the thalamus or the sensory input in the brain. So how can we do that? So we can genetically silence layer 6B neurons selectively if we get rid of a protein which is involved in presynaptic vesicular release. And this protein is SNAP25, which was discovered by Michael Wilson. And without SNAP25, these synaptic vesicles cannot dock and get released in a regulated fashion. You still have some spontaneous vesicular release, but this is much less than with the action potential coupled synaptic vesicular release. So that's what we did. We did a conditional SNAP25 knockout. We also did many other conditional knockouts in layer 5, layer 6, and layer 6B. I just show you the results now for layer 6B. And basically, this is a chronic silencing of these cell populations. So we silence them from birth. Then it will have consequences on some of the fibers will degenerate, some of the synapses will degenerate later on in these cell populations, but this is not until later in life. And then we have never seen increased cell death until around 8 months in layer 6B silenced animals, but we could see that in layer 5 and even in 6 silenced animals. So we have a chronic silencing, which has a long-lasting effect on the maintenance of the synapses and the maintenance of the myelin. But initially, the development is pretty normal in these animals. So how will this change the behavior of the animals? So we expected that this kind of silencing will have an effect on the sleep rhythm or circadian rhythm of the animals in layer 6B. But actually, I was wrong, because this did not lead to that. So we produced these conditional layer 6B knockout animals, and then they had very little behavioral change in what I expected. So there was no change in the circadian rhythms of these animals. They responded to jet lag, so when we give them a phase advance, just as well as the normal animals. If we put them in constant darkness, they have the same kind of pattern of activity, and also their locomotor activity was not different from the normal animals. We also looked at their sleep activity, and there was no change whatsoever. If we do a similar experiment in layer 5, you can reduce the sleep with 3 hours. We just recently published that. And there is a very strong phenotype in layer 5. But in layer 6B, where I expected this, there was nothing. However, these animals have much less anxiety. So if you put them into elevated plus maze, then they go out to these open arms, which a normal mouse would find more stressful. Or if you put them in a light dark box, they spend quite a bit of more time in the light part of this box, where normal animals would go to the dark. We also did some behavioral studies in a Y maze, and this Y maze basically, if you block off one arm, and then these animals can explore only one arm, when you reopen this blocked arm, then 80% of the animals will go to the novel arm, but not the DRD1A Cree SNAP25 animals, so the conditional layer 6B silenced animals. They go 50-50 after this. So they have reduced novelty preference if we silence layer 6B. So we are running other tests, behavioral tests on these animals, but it does look like that silencing the cells, which are the major targets of orexin in layer 6B, will have an important change in behavior regarding anxiety. And it has been known that orexin is involved in fear and anxiety, and there are lots of possibilities to use that even in the clinic. So we are particularly interested in interfering with the cortical targets of orexin, the layer 6B neurons. So there are lots of questions which we need to check to formulate this hypothesis further. And the first question was that, are the DRD1A3 positive layer 6B neurons orexin sensitive? And indeed they are. Now we know the answer to that. We are currently doing in vivo recordings from layer 6B during state transitions, and we want to see whether this cell layer is active during these state transitions. Layer 6B is a tricky area to record from because it's very thin. So we have to use all sorts of tricks to get this data. But now, hopefully we will get some data on this. We are also very interested with Vlad Vyazovsky, who is my major collaborator on this, is whether there is a local sleep problem here. Vlad in Tononi, they discovered the local sleep in rodent model, and we want to see whether this is affected in the layer 6B activated or inactivated models. And finally, the biggest question is whether the human interstitial white matter cells, whether they show similar sensitivity to orexin, or whether they have long distance projections or local projections. We know very little about these cells, although you can see them in all mammals in certain percentages. And if you add them up, they are just as big as the visual thalamus. So this is a substantial population of cells. So these are the outstanding questions. But I just want to leave you with this message that, you know, although these cells, these layer 6B cells, they're very important during development, and the majority of these cells will die off from the brain. The remnants of these cells are somehow repurposed. And these repurposed cells, they can have quite an important role in functional states, in state control in our brain. So they have a very important role how we process sensory information. So as you know, you get 10 times more input to your thalamus from your own cortex than from the periphery. And this output from the cortex is regulating your sensory input to your cortex. So most of the time you are listening to your own brain, and you decide what you pay attention to. And that's why I think these layer 6B projections could have a very important role in regulating these events. So, and that's why we will study them in more detail. And also the dream is to link them to some of these cognitive dysfunctions, which you see during brain developmental abnormalities when you have increased interstitial white matter cells, which survive in the brain. So we want to link these cells in human pathology into some of these conditions, and we want to understand what is the role of these cells in the manifestation of these conditions. So I am very grateful to my laboratory and past and present and laboratory members, and also all the collaborators and the funders. And I'm also very grateful to all of you for your attention.
Dr. Molnár, thank you for this excellent lecture. It's amazing to know how much detail goes into these studies and know how complex the brain is from the beginning until the end of life. So it's amazing. I assume we have 5 minutes for questions. Is that correct? Okay. So one of the questions that came up was how different cells of different origin receive the signal to migrate to the subplate. You showed at the beginning that we have different cells from different origins migrating to the developing cortex. Do we know the exact signal that guides them there? Because they're different, right? So,
very good question. I think it's a fundamental issue, that how this initially just a very thin cell layer, which is will contain most of the marginal zone and subplate cells. How do they know which cell is going to the marginal zone and which one will stay in the subplate? And later on, also subplate is a very important corridor for the migrating GABAergic neurons. But when you look at the adult, then in fact, you have less interneurons in the subplate than in layer 6B in the mouse than in any other cell layer. With Anna Horder-Zabadis, and we looked at this, and we were so surprised to see that during development, you have most of the GABAergic cells migrating through this zone. Yet, when you actually look at these compartments later on in the adult, you have actually the least density of GABAergic interneurons. So it's a dynamic structure, and the composition of the subplate is changing over time. And subplate is actually regulating migration of the cells. It's Chiaki Maruyama in Tokyo who discovered that subplate is forming transient synapses with the migrating cells when they go through the subplate, and they regulate the migration of these cortical plate neurons to the brain. It has only been shown in rodents, so it would be very interesting to see whether the same is true in a larger brain, in a macaque or in human. But according to Chiaki, the subplate is sorting out some of the migration to the cortical plate. So they are quite important in that as well.
I see. Actually, one of the questions that I had is whether when this death happens or when it doesn't happen, does this affect the GABAergic neuron migration? Because it happens in different stages, right? And quite early in development, if I remember correctly. So does that change in pathological conditions when we have more cells?
You addressed again a very, very important question. So if we change the composition or activity of subplate neurons, how would it change the interneuron distribution in the cortex? And I must say that we haven't done, or I don't know whether anybody has done such experiments, but we have done an experiment where we actually changed the activity of layer 5 projection neurons. So it's not subplate, it's layer 5. And what we noticed that it had a huge effect on the distribution of microglia and also the distribution of interneurons. So basically what you do with the pyramidal cells, it will have an effect on the distribution of the GABAergic interneurons. And you are absolutely right. It would be very interesting to see if you silence layer 6B, how this is changing the interneuron distribution in the rest of the cortex. And we haven't done that so far.
But somehow the number, I guess, it's inversely correlated with the interneurons, or is it grammically? If I remember, you said in the mouse, at the later stage, you have less interneurons despite having them migrating through.
So you could actually, all our systems are chronic silencing. So we basically, the Cree recombination occurs around birth, and after that you have silenced layer 6B or 6 or 5, depending on which condition or knockout. Of course, you can use chemogenetics to activate them in a different time frame. So you could use inhibitory excitatory dread systems and then look at the interneurons. And this has been done for layer 5 by Oscar Marin and others for the GABA cells. But I completely agree with you. It would be equally important to see it for another cell population like layer 6b, how this would impact on the interneuron and whether a different population would have a change.
I see. So another question is, do we know how the preferential cell death takes place, the mechanism? Because some cells, I guess, the remnant cells stay back. What differentiates them? Is it the position or the random synaptic formation that, if we can call it random, that takes place?
Very good question again. So the question is, how could we, so how do the subplate cells know which one should go and which one should stay? Yeah, and as I think this is a fundamental issue, because that would link these interstitial white matter cell changes to the human pathology as well. So that's why it would be really good to know this. So we have a couple of markers which delineate subplate cells which will survive, and this is CTGF, NUR1, LPAR1, and other markers. But we don't have markers for the subplate cells which will die, and that would be also important to pull out. Now, how could we change the cell death in subplate? And I did some experiments long time ago, I think more than 25 years ago, when I cut the internal capsule at embryonic stages in mice. In fact, I did it in Pittsburgh with Ray Lund. I think 20 or 30 years ago. And basically we injected thymidine at embryonic 11, and then we did the manipulation of the internal capsule. And when we looked at the subplate, the heavily labeled subplate cells, birth dated cells on the two sides of the brain, with the internal capsule cut and the normal. And what was interesting is that more subplate cells survived in the region where you had the cut. Okay. And also when you put a tracer into that hemisphere, you had a large number of subplate cells reaching the tracer injection. So either they maintained or developed projections in that isolated cortex, and they stayed there longer. So maybe the survival of the subplate cells dependent on the development of the cortex. And if the cortical development is arrested, then you have more subplate cells surviving because they were not kicked out of the circuit, or they, so whatever is causing their death did not occur. So it would be very interesting to see whether it's actually the activity, connections, or timing, which is important for these cells to die off. And also it would be really interesting to know exactly which interstitial white matter cells survive in human in schizophrenia, because there are lots of different subplate cell populations. Is it a certain type which is remaining?
I see. So
these are key questions.
Yeah. And do we have any clues on the orexin part in terms of the human pathology now? And how, I guess it's kind of difficult to address that part, as you mentioned, with the interstitial cells, because you need samples, and it's not it's not easy to do that. Do organotypic, do you use, or can you use organoids or organotypic slices to address this kind of issues, if that's possible?
So if you look at the single cell transcriptomic data from developing human brains, Kodoshevich's lab in Copenhagen, or Ellen Brain or Arnold Krickstein, actually you can see that these cells are actually orexin sensitive. So you have a cluster of interstitial white matter cells or subplate cells, if you dissect them before you do the single cell transcriptomics, that actually you have a hypocretin receptor expressing orexin sensitive cell population within subplate, coexisting with the expression of CTGF, complexin 3, which mark this population. So, but what we don't know is whether in schizophrenia you have, again, orexin sensitive interstitial white matter cells, and whether this particular cell type changed its density or distribution. So we have to work more on that. So there is a link, but we don't know the pathology yet. You know how it is manifesting.
Okay. Well, I guess it's good that there are a lot of open questions and challenges, because you have to find a way to overcome them. But I guess that's a challenge on its own. So, we don't have any questions, any other questions. So we wouldn't hold you anymore. Thank you again for the excellent drive-through development that you gave us tonight, and it was a real honor to have you in our lecture series, Dr. Molnár.
Thank you very much. The honor was all mine. Thank you very much.
Thank you. Have a good night.
Thanks. Bye-bye.